Okay, so we're gonna get started. So please join me in welcoming Dr. Matthew Brown, a postdoctoral associate at Rutgers University and a leading entomology specializing in integrated pest management for photographs and ornamental systems. With over 7 years of applied research experience, Dr. Brown has developed innovative solutions for managing challenging pests, including the Bermuda grass mite and the annual bluegrass wheel. He holds a PhD in entomology from Clemson University, where his work focused on combining cultural and chemical strategies to address turfgrass pest infestations. His research has resulted in numerous peer-reviewed publications, extension materials, and stakeholder-driven outreach programs. Beyond the lab, Dr. Brown is an active science communicator, mentor, and advocate for early career scientists. His work continues to bridge the gap between science and practical applications in landscapes, golf courses, and sport turf systems. Please welcome Dr. Matthew Brown.
Hello, thank you everyone. Thank you for coming. Um, Today I will be telling you about, can you hear me all right there? Or does this mic work? Oh, the mic doesn't work. Oh, okay. So, yeah, thank you everyone for coming out. Today I'll be telling you about some research I've done here, a few different projects that mostly aren't really related to each other. First, here I'll tell you about some research I did in my PhD, where we looked at integrating the use of tomato cultivars that were developed for resistance to arthropods with biological control using commercially available predators. And here, this project had two parts, one where we worked with two-spotted spider mite and another one that worked with thrips. So two-spotted spider mite's a pest of tomatoes, but also a lot of plants, especially greenhouses. And so as part of this project where they developed these different tomato lines to produce more of acyl sugars. And so that was supposed to help make them resistant to different arthropods on tomatoes. Acyl sugars are these metabolites that are produced in droplets from the hairs of plant leaves on tomatoes. And in prior research, they didn't show any toxicity, but instead they deterred arthropods from feeding or laying eggs. And so here they had developed 4 different resistant tomato lines. And in these experiments, we were testing these different resistant tomato lines along with this commercial cultivar that we were considering kind of the baseline of susceptible cultivars that are available. And prior research that one of the postdocs in our lab had done was looking at resistance of these cultivars to spider mites, and where we found that the spider mites preferred the commercial cultivar over all of these resistant lines, and that they preferred one of these cultivars, CU07, to most of the other resistant lines. And those were lab experiments. Then we also did kind of the same thing in a greenhouse with whole plants to see if spider mites, how they built up populations on these resistant lines, and found similar results where they built up much higher populations on the commercial cultivar. And then one of the other resistant lines, they built up some populations. And so part of this project is we wanted to see, could we use biological control with those resistant tomato lines that they had developed, specifically targeting organic field production where pesticides aren't available. So really host resistance and biocontrol being two of the major strategies available to us to use in those systems. And here we looked at 4 different predators, 3 predatory mites, and a ladybug. And, we thought that basically these natural enemies are going to be exposed directly to the acyl sugars, just like the spider mites are, because they're just on the leaves. And so it's not really to do with feeding on the leaf. It's just that if you walk on the leaf, you're going to be exposed to them, or whatever they're doing to deter the spider mites, that the predators are going to be exposed to the same properties. And so here we did partly a lab experiment where we looked at a commercial cultivar and the least resistant tomato line, where we did choice experiments in petri dishes. We put spider mite eggs on there to measure how many eggs the predators were consuming, and then we released 5 predators of each of those 4 different commercially available predators. So predators that you could buy and then release on tomatoes in the field. And what we found was that the predatory mites preferred the commercial cultivar, not so much the ladybug, and that predators consumed more eggs on the commercial cultivar, including the ladybug. But the predatory mites and the ladybug species, they all consume more of the spider mite eggs, so predation was much higher on this commercial cultivar compared to these resistant lines, indicating that, yeah, these resistant lines they were interfering with biological control of spider mites in tomatoes. And so then we did also these whole plant experiments, very similar, except we didn't use the ladybug species here, but basically where we infested these plants with spider mites, and then we introduced predators and collected leaves throughout to see how much predator populations built up and if they were able to have an effect on spider mite densities. So here, what we found was that here we're looking at pest densities, really just on the commercial cultivar, and we found that, okay, whenever we introduced the 3 treatments with predatory mites compared to the check treatment where we didn't release any predatory mites, that they were reducing pest densities. And that Phytocoelus persimilis was the only predatory mite that reduced pest densities on this resistant line, even though that may have been just because of the fact that the pest densities were so low on the resistant line anyways. And overall, that resistance had a stronger effect here than predators, even though that may have been mostly because of the fact that our setup, that we were infesting these plants with spider mites beforehand, and the predators we just released once at the beginning of the experiment, and then waited 3 weeks. And also that predator populations were lower on the resistant line, even though that may have just been because of the fact that they had lower pest densities, so that's less of a food source to maintain populations. But overall, the important point from this research was that we identified that these resistant tomato lines were interfering with biological control using these commercially available predators. That was really the important point. That was mostly from our lab experiments. The whole plant kind of confirmed that, even though, again, that could have been because of the fact that the pest densities were so low on the resistant line anyways, that was why biological control wasn't having much of an effect and predator populations weren't able to maintain as high. That resistance was greater than biological control, and that we identified these 2 predators that seemed to do the best on resistant tomato, even though still they were having much less of an effect on resistant tomato rather than the commercial cultivars. Yes. Okay. Where is it here? Click on this here. Oh, okay. I see. Okay. So this next project we did was I did here, postdoc here at Rutgers, where they were already doing this project that I kind of joined on when I came in, where we were looking at see if I can go. Okay. Where we were looking at converting this pest, annual bluegrass weevil, that is one of the major pests on golf courses in this area. We're looking at converting this pest into a beneficial, basically, of a biological control agent of a weed in turfgrass. And so, to give you a bit of background about this pest, annual bluegrass weevil, it's, again, one of the major pests in this area on golf courses, that it mostly targets short mown turf, where the adults, they emerge from overwintering sites, and then they migrate to short mown turf. They lay eggs. The young larvae, they feed within the stems of turf grass, and then they emerge as older larvae to feed at the crowns of turf grasses, and so those are the most damaging stage, and that's where we're going to see most of the damage in this area in late May, early June. as the older larvae. See if okay. So as far as the plants relevant in this system, most of the time they're going to be a problem on creeping bentgrass turf, on fairways, on golf courses where it is short mown. And creeping bentgrass, it's the turf grass that they're trying to grow. So it's the high value grass that these managers on golf courses are intending to grow, right? It's bred for certain characteristics that are favorable for turf on golf courses. But there's also this other plant, annual bluegrass, that is a weed, but basically it's so persistent and so hard to get rid of that it's constantly there. So they can't exactly totally get rid of it. So a lot of times you're going to have turf that is mixed with creeping bentgrass and annual bluegrass. And not only is it there, but sometimes it's in such high amounts, it might even be like half of the turf, that if all of this weed dies, you're still going to have, half of your grass is going to be dead. And so that's still going to be a problem because it's going to look bad. It's going to have worse functional characteristics. And so ultimately, they do have to maintain this weed too. It actually has higher maintenance requirements. It requires more water, more fertilizer, but they do have to maintain it because it is there in the turf. And if it all dies, and it's a huge percentage of the turf, it's going to cause problems. So that's kind of where this project came from, was the fact that annual bluegrass weevil, as I talked about, this pest that's viewed as a pest, currently managers on golf courses are using insecticides to try to kill this weevil because of the fact that it's a major pest. But one of the interesting things about it, as its name suggests, annual bluegrass weevil, it actually prefers to eat this weed. We've done greenhouse experiments. It prefers to lay eggs in annual bluegrass. It builds up higher larval populations. And also annual bluegrass has lower tolerance. So it has more damage when weevils feed on it. So this weevil preferentially damages the weed, annual bluegrass, compared to the high value turf grass that managers are wanting to grow. And so that's where this project came from, is thinking about trying to use this pest instead as a beneficial, as a biological control agent of the weed, because it preferentially damages it. So basically just not using insecticides to kill the weevil and instead letting it go and preferentially damage this weed. And so some of this work was already done where they looked at, okay, if you didn't use insecticides to kill this weevil, could it come out and reduce levels of this weed, and found that yes, that was the case. But that one of the major problems with this strategy is that, kind of what I talked about at the beginning, that if you have if the turf comprises a lot of annual bluegrass, so if like half of the turf is annual bluegrass and you let this weevil come and damage all of it, then that's going to still lead to a lot of damage, and they're not going to want that. So we consider this one of the major factors in considering whether this strategy of letting the weevil act as a biological control agent of the weed would be how much of the turf is made up of the weed. That basically, once it's so much, it's going to be a strategy that isn't really feasible because it's going to lead to so much damage. And so we did some field experiments where we looked at a few different insecticide programs, and that was basically preventive. That's just to totally prevent weevil damage. That's like what's currently used by managers on golf courses, basically for pest control of this weevil. No insecticide, where we're just not using any of those insecticides, or also a threshold-based one where we're waiting to see if weevil damage shows up and then using insecticides. And then another factor was weed cover. How much of the turf was made up of this annual bluegrass, which we were actually able to manipulate, so we could have treatments of like 0%, 10%, 25%, and 50% of the turf made up of annual bluegrass, of the weed. And then also throughout the experiment, we measured basically how much of the turf was made up of that weed. So that was a treatment, but then also what we measured throughout the experiment, because it's changing, again, especially based on whether we're using insecticides or not and whether the weevil is coming in to damage it. That's what we figured. And then also we looked at turf quality throughout the experiment, essentially as a measure of weevil damage, even though it really is referring to quality as a whole, especially differences between insecticide programs. We're attributing mostly to the fact that the weevil's coming in and damaging the turf, and so that's what's leading to changes in quality between those treatments. So here we're looking at weed cover. So this is the percentage of the turf that this weed is made up of, or percentage of the turf that has this annual bluegrass, has this weed. And on the left graph, it's showing basically our insecticide program treatments, which we saw basically what we expected, that when we used this preventive insecticide program, we had higher levels of the weed, mostly in the second year here. That's kind of the important part of this data here, is that, yeah, the preventive insecticide, whenever we use insecticides to kill the weevil, that's leading to more weeds. So insecticides are interfering with weed control, or you could think of it that if we don't use insecticides, then we're actually getting more weed control. And this is basically just the repeated experiment of that, where we had this over 2 years, and where again we saw similar results, where whenever we used insecticides, it interfered with weed control. We had higher levels of this weed, which again, that was something that we kind of already knew from previous research. But then we also wanted to look at turf quality, which essentially we're just considering kind of a measure of damage. And so here we saw an interaction between these two factors, between the insecticide program, which are the lines here, and then the different graphs are the different levels of the weed that we started with. And what we found is, largely what we expected, that whenever we use the insecticide program, we, especially at higher levels of the weed, that was protecting from damage. So that, again, that's why people are using insecticides in the first place. But that whenever we had lower levels of the weed, really typically there wasn't much difference even whenever we did use insecticides. So insecticides weren't even really so much needed at lower levels of the weed. But whenever we had higher levels of the weed, that, it was leading to unacceptable levels of damage, which here we kind of considered these gray dotted lines in turf research. That's what they assign this rating as the minimum acceptable quality. So basically, if it goes below that level, it's considered unacceptable. And so in this case, whenever we didn't use insecticides, yeah, it did reach that level, but most of the time it recovered even still within a month. Especially without insecticides, it recovered in a month. Whenever we used the threshold program, it basically sped up recovery. So whenever we applied, when we saw damage, that kind of sped up recovery and still used less insecticides than just totally using insecticides, no matter whether there was weevil damage or not. And here, this is basically just where we repeated. We didn't really see the same interaction between the factors, but we did see the same effects where, insecticide program, yeah, was protecting from weevil damage. But here in the second experiment, we didn't actually see ever where it really reached unacceptable levels. So even though insecticides were protecting the turf quality, that we never saw where these strategies actually led to that unacceptable damage. And again, that was kind of the most important part of this, is that we were showing that, yeah, insecticides that we're using to try to kill this pest, weevil, are actually interfering with weed control. And so if you don't use insecticides or use less insecticides, then you're saving the cost of using insecticides against this pest. And also you're getting this benefit of weed control, and saving costs there as far as whatever you would use to control the weed otherwise, herbicides probably. And what we were mostly wanting to do here was try to identify some level of weed cover at which this strategy was feasible. Basically, we weren't going to have unacceptable levels of damage. And so in this case, it was basically under 35%. When we had less than 35% of annual bluegrass in the turf, that this strategy of not using insecticides was feasible. And that basically the turf grass actually recovered relatively quickly, right? That whenever we didn't use insecticides, it took about a month. Whenever we used that threshold program, it sped it up to about 2 weeks. So even when we didn't use insecticides and it did lead to unacceptable damage, it recovered fairly quickly, where there was no difference whether we used insecticides or not. So, I mean, that kind of can depend on the golf course and the manager, that, people are going to have different levels of tolerance in terms of how much damage they're willing to accept. And so, this is kind of information that can help them decide that. But some of them may be able to even, deal with unacceptable levels if it's going to recover, or, some of them may have extremely low tolerance and not want to use this strategy at all because of the fact that it is still going to, unless it's at really low levels, like 10%, it's still going to lead to some level of damage between using insecticides and not using insecticides. Okay. And then, yes. Okay. I mean, mostly it's based on, research and like weed science as far as what they do there. As far as I know, the recommendation is there's like a plant growth regulator that is considered the standard for helping to get rid of annual bluegrass. It's kind of the same problem as we were talking about with the weevil. If you have high levels of annual bluegrass, if you just kill the annual bluegrass, that's still going to be a problem. So even in weed control otherwise, they use a plant growth regulator that basically makes it where the annual bluegrass doesn't grow as much and creeping bentgrass isn't as affected by it. So it can grow into the areas where annual bluegrass isn't growing. And so it kind of slowly, over time, the high value turf takes over the weed. And so that's usually what they suggest as a plant growth regulator. But there are also are herbicides that are available. So, but primarily, I would guess a plant growth regulator would be better in those cases where you have higher levels, maybe to then get it down to this lower level where you could use this strategy of just letting the weevils take out the weed. So for these final two projects, they're both kind of to do with looking at effects of different things in turf systems on entomopathogenic nematodes. Also, this project looking at the effects of fungicides on nematodes and insecticides against different turf pests. Again, I talked about annual bluegrass weevil before. Also, there is this other pest, white grubs, which is a very common pest in turf all across the U.S., where it feeds on the roots underground. Black cutworm, another common pest, really more so in a specific context of on creeping bentgrass greens, where it's a nocturnal insect. It burrows in the ground during the day, and then at night comes out to feed on the above ground plant parts. But all three of these insect pests are, good targets and have been researched previously for targeting with entomopathogenic nematodes, which there are products available in turf grass to use for biological control of turf grass pests, especially ones like the ones I just mentioned that are either live in or near the soil. And so here, this project, we wanted to look at the effect of fungicides on insects in turf systems. Partly, fungicides are very commonly used in turf grass to manage plant diseases. They're much more so used than insecticides. And that there's these research from other systems showing that fungicides, while they don't exactly act like insecticides on insects, that they do have effects on insects, whether that is causing increased mortality. Even in turf grass, that some fungicides have been shown to increase mortality of early instar white grubs, or also increase susceptibility of annual bluegrass weevil to insecticides. And so, and also in other systems that they delay development, or that they affect like, certain enzymes that are involved in detoxification or immune response of insects. And so with this project, we wanted to look at, that fungicides may be able to have some synergistic response. They may be able to weaken insects or interfere with their response to toxins, in that case being insecticides, or pathogens, entomopathogenic nematodes. So we had really a series of experiments that I'll kind of try to go through quickly here with all three of these different turf grass pest insects of lab and field experiments. Where here we did ones with adult weevils in a lab with petri dishes. We fed them clippings. We applied four different fungicides at two different rates, and then applied nematodes and basically just looked at how that affected adult weevil survival. And so this graph kind of has a lot, so I'll try to explain it, especially because there's going to be a few graphs that look a little bit like this. So basically, in the top graph there, we have fungicide alone. So it's basically where we just had fungicides. And the bottom graph is where we had a combination of fungicides and nematodes. Then there's two different rates for each fungicide, with either the kind of hatch pattern or the solid bars. And then in the second graph, the letters are indicating, what you'd expect, significant differences between the different treatments. But then we also have these asterisks that are indicating significant synergism or antagonism between the fungicides and in this case, nematodes, but could be insecticides too in some future graphs. And then there's also a number, which is indicating, if it's positive, basically that's indicating synergism. And if it's negative, that's indicating antagonism. And so what that's meaning is that, synergism is meaning that basically, so there's like some effect of nematodes, there's some effect of the fungicide itself. If you add those together, that's basically what we're considering an additive response, whereas synergism is saying there's some boosted response beyond just adding those two effects together. Antagonism meaning these treatments might be interacting in a way that they're interfering with each other, and so there's even less of a response than adding them together. In this case, all of them had some positive response that was greater than additive, even though they weren't all significantly synergistic, only the ones with asterisks next to them. But we did ultimately find that some of these fungicides were synergistic with nematodes. That's kind of the important part of that. Then we did a field experiment that really wasn't so related to the lab experiment here with weevils, because of the fact we did this field experiment first. Actually, here we're targeting larvae. In the lab experiment, I mentioned we're targeting adult weevils. Mostly in turf grass management, we're targeting larvae with nematodes because they're more susceptible. Also, they're the damaging stage. And so that's what we started with here, was doing this field experiment with looking at larvae and basically these combinations of fungicides and nematode. But here in this research, we didn't really find any effect of fungicide that it was synergizing with nematodes or that it was increasing the effect of nematodes. And so basically that's why we kind of switched in the lab experiment to looking at adults instead, because we didn't find this effect with larvae. So instead we thought adults are less susceptible, but that they may have this effect that we're not seeing in the larvae. Then we also looked at, again, this other pest, black cutworm, in the lab, where we looked at so many different fungicides that are commonly used in turf in the context where they would be relevant with black cutworm. We looked at, here we looked at more so how they synergize not only with nematodes, but also with bifenthrin, an insecticide that's used to manage black cutworm. Then looked at how that affected survival, development, time to pupation, basically using that as an indicator of development, and then growth. Ultimately here, this graph is kind of set up in a similar way to the other one. The top one is the fungicide alone. There wasn't any fungicides that showed any effect by themselves compared to the control. But we did see a lot of synergism with a lot of our fungicides at different rates here, where there was synergism between the fungicide and the nematodes, even though not all of them were actually different than the nematodes alone. Then again, we had another part of this experiment where we looked at synergism between the fungicides and an insecticide on black cutworm. And we didn't see as much synergism, but we did see some of the fungicides had some synergistic response with bifenthrin, where they increased they increased mortality in the combination treatments compared to using the insecticide alone. Then again, we also looked at some of these other measures of just the fungicides themselves to see if they were affecting development or growth. We did see that most of the fungicides delayed development, that it took them longer to pupate, but it didn't affect growth. Then from those lab studies, we used that information to develop a field experiment in which we would test some of those that showed synergism in the lab in the field. where we looked at pyraclostrobin and fluazinam with nematodes and insecticides. We released black cutworms and plots in the field, and then we used a soap flush basically to bring the cutworms up and count them. So here, looking at nematodes, the hatched is the combination treatments with nematodes. The solid bars are the ones without nematodes, just the fungicides. And here we found that fluazinam did have a synergistic interaction with nematodes. Pyraclostrobin didn't, but it did still, the combination treatment reduced densities below whenever we just use nematodes, because of the fact it seemed like pyraclostrobin itself alone had some effect on densities of black cutworms. Even though this data is a little bit more preliminary, we only have one experiment I'm showing here, and so there's a lot more variability. And then we also looked again at bifenthrin insecticide in the field, and we didn't really see any effect with synergism with either a fungicide, even though that might have been because we had low densities overall. In our control, you can see we released 40 in each plot, but we really only recovered 4 from each plot. So, that's kind of been up and down. We usually don't recover much, but I think previous experiments we had recovered around 10 after a few days. And then once we use the insecticide too, that reduced density so far that we were covering on average like about 1. So that may be why we didn't really see any effect. Then again, we also did some of these same experiments with white grubs, where we had these lab experiments where we were combining fungicides with nematodes and insecticides, looking at effect on white grub survival. And here, with fungicides and nematodes, we did find pyraclostrobin had a slight synergistic response with fungicides and nematodes, even though the treatment wasn't greater than nematodes alone. So there was a synergistic interaction, but it wasn't actually, didn't actually have higher mortality overall than using nematodes alone. And we did see an antagonistic interaction from one of the fungicides. Then looking at insecticides, we looked at 2 insecticides here, and we now saw that kind of depending on the fungicide and the insecticide at high rates, we did see these synergistic interactions where some of the fungicides had synergistic interaction with one insecticide and some with the other. And so then we also looked at, again, how the fungicides themselves just directly affected white grub weight. We didn't really see much effect there. And so we use this to inform doing field experiments with some of these fungicides that did show synergistic interactions with the insecticides. And here we found that in the case of trichlorophone, which was one of the insecticides, we really didn't see any synergism there with the one fungicide that we tested. But the other insecticide, clothianidin, we did see synergistic interactions. And kind of like with black cutworm, pyraclostrobin itself seemed to have an effect on white grub densities alone. Even though we didn't see those in the lab experiments, we did see that in the field that it had an effect on white grub densities, and that both of the combination treatments had lower densities than using nematodes or using insecticide here alone. And so that was kind of a lot of where we did, I guess, lab and field experiments, which each of these different pests. So I kind of summarize them here, is that really a lot of the fungicides we did see some synergism with, even though mostly that was with black cutworm, where we tested a lot of these. It did seem like it was especially susceptible to these synergistic interactions, especially with nematodes. We didn't really always see the same effect in the field. Sometimes we did clothianidin with white grubs, and then nematodes with black cutworm. And so that could have been in the case of the weevil, because we were looking at different stages, adult versus larvae. And black cutworm could have been because of the fact that we didn't really have great numbers in the field. But otherwise, yeah, we mostly didn't see that. Sometimes it translated to the field, the lab experiments did, but sometimes not. So also, Again, we found that rarely fungicides had some effect alone. Um, in the field or mostly in the lab, we really didn't, other than that they were delaying development of black cutworm. There was one fungicide that antagonized with nematodes, with white grubs in the lab. And so why this information is important in turf grass is because of the fact, again, that fungicides are already being used in turf grass a lot. And so they're already, we already have these fungicide applications going on. And so that really, you can use this information to potentially increase nematode control of insects with nematodes and insecticides without actually using any more chemicals or having any higher cost, by basically just timing your nematode insecticide applications with fungicide applications that are already going on. And, we tried to use fungicides that were relevant, that turf grass managers would be using at times of the season when these same pests were also relevant. So that's why we tested a lot more with black cutworm, because at the time that black cutworm is a pest that you're going to want to control, there's a lot more fungicides available. Also, too, you don't necessarily have to get it into the soil to contact black cutworm in the same way that you do with grubs and weevils. And that ultimately, these synergistic interactions did show up with all of the pests that we tested, all of these different turf grass. So it says maybe they have some kind of broad effect that can be taken advantage of. And that even though the ABW larvae, we didn't see any effect, we did see an effect with adult ABW. And there's previous research that's showing some of these fungicides increase susceptibility of adult weevils to insecticides, even though that was mostly thinking about it from the perspective of weevils that are resistant to insecticides. And so then this last project I will tell you about is kind of a little bit along similar lines of looking at some factor that's used in turf grass and looking at its effects on biological control of turf grass pests with nematodes, mostly in this case, plant species or cultivar. And so again, we're working with, in this project, cutworms and white grubs, not weevils so much. And just to give you some, a little bit of background information about plants and turf grass, mostly in this area, we're talking about cool season turf grasses on golf courses and lawns. Here I worked with creeping bentgrass, tall fescue, perennial ryegrass, and Kentucky bluegrass, all cool season turf grasses. Then also there are weeds present in some systems. Annual bluegrass I talked about before in one of the other projects, and white clover, also a common weed in turf grass. And for example, that these plants are a lot of times going to show up in different places on a golf course, where on a green you're more likely to see creeping bentgrass, right? That there's turf grasses, generally they're bred in a way for characteristics that are suitable, that managers on golf courses want for aesthetic characteristics, or maybe that creeping bentgrass can be mowed a lot lower than other plants. Whereas on a fairway, and for example, a green also too, they have much higher maintenance inputs. They're going to be putting in much more water, fertilizer, and pesticides. And like, for example, weeds, you're typically not going to see on a green because of the fact that at least the manager doesn't want to see them on the green, right? So they're going to put in a lot of inputs to try to make sure that there are no weeds on greens. Fairways they're kind of intermediate between greens and roughs. You're probably still going to mostly see a monoculture of one turf grass, probably creeping bentgrass. Could be other turf grasses though, and not so many weeds. Whereas a rough kind of depends on the golf course and what the manager wants, but you're much more likely to see different turf grasses there together, and also weeds, because there's not as high of maintenance inputs to try to kill weeds, because partly also too, greens are going to make up a much smaller portion of a golf course, than fairways, and then fairways are going to make up a slightly smaller portion than roughs. And so why we think plants could influence biological control with nematodes in turf grass, partly based on previous literature from other systems, that For example, root systems have been shown to affect nematodes, especially nematode movement. That larger root systems make it harder for nematodes to move and to be able to infect insects. Plant chemistry could affect the insects themselves, their physiology, change their immune response. Also, they might be able to sequester plant chemicals from the plants that then affect how the nematodes are able to infect, or also nematode mortality within the host. Plant chemistry could also affect nematodes themselves. Roots release volatiles that are potentially going to attract nematodes to the roots. Also, they might release chemicals into the soil that provide a more favorable soil environment for nematodes. Also, plants might modify insect behavior, especially resistant plants. Insects might move around a lot more to try to find places to feed, and so that might make them more likely to for nematodes to contact them. And so with this project, we were basically just looking at, kind of similar to the first project, where we're looking at different turfgrass species, weed species, and different cultivars of one of the turf grasses to see how that influenced EPN performance against black cutworms and white grubs. And so we did a lab experiment here with black cutworm, where we grew these 10 different plants, a couple weed species, 4 turf grasses. One of the turf grasses, we did 5 different cultivars, and fed the cutworms these plants for a while, and then applied nematodes and looked at how that affected cutworm survival, cutworm mortality. So ultimately here, again, the top graph is showing basically just them being fed the plants. Not there's no nematodes being applied, and then also the bottom graph is showing basically where they were fed the plants, and then nematodes were applied to those petri dishes. And what we found mostly was that there was some resistance in some of the plants. Kentucky bluegrass is resistant to black cutworm. That's something that there's already been research on showing that Kentucky bluegrass is resistant to black cutworm, so that's something that we expected. White clover was slightly resistant. But then also, too, that those same plants, Kentucky bluegrass and white clover, had the highest mortality with EPN as well, with nematodes as well, which maybe because of the fact that they have resistance, especially in Kentucky bluegrass, it may have just been some effect that they have resistance, and so then once you add nematodes, that also has some further effect that so that results in it having higher mortality than the other turf grasses. White clover, again, it could be that it has some resistance too, or that it's also having some positive effect on increasing mortality with nematodes. So then we also looked at different cultivars of creeping bentgrass with black cutworms. We found that ultimately we didn't really see any differences in mortality whenever we just had the plants, the different cultivars alone. So there wasn't really any resistance with these plants, but that some of them, there were differences in whenever we combined that with nematodes, and that there were some differences in the cultivars. In this case, mostly it seemed like Declaration and Pin A1 had some higher mortality than some of the other cultivars, but it was kind of a little bit messy as far as maybe one cultivar had more than a few, another cultivar had more than one. We basically took that same idea and carried it to the greenhouse, where we looked at turf grass grown in pots, where we looked at these 5 different cultivars with black cutworms, because of the fact that, again, black cutworm management is relevant on creeping bentgrass greens. So we didn't really look at the other species here. We just looked at these 5 different cultivars because of the fact that's mostly where management with black cutworm is relevant. And then looked at it with EPN, and then also had infested pots and non-infested pots, so that we could see maybe if there was some aspect of tolerance here too, where black cutworm is maybe eating more of one grass, or the grass is able to recover in a way to make up for whatever eating the black cutworm is doing. So here we saw actually fairly similar results to the lab experiment, where none of these cultivars had any resistance to black cutworm in the without nematode, the top graph. there weren't any differences, but that whenever we applied nematodes to cutworms that were fed these different cultivars, there were differences, even though it was, again, kind of messy, but it seemed like mostly declaration, pin A1, even though I think they kind of swapped from the lab experiment, they were the two that had the highest mortality in this greenhouse experiment as well. So showing that there was some differences in how these cultivars affected nematode performance, kind of the important part there. Then again, we also looked at weight, black cutworm weight, how these to kind of look further at resistance. We didn't find that they had any effect on black cutworm weight. They did have an effect on reduction in shoot weight. That's basically just our difference between our infested controls and our non-infested controls. And so that there was some difference there where it seemed like they kind of separated into two groups where there was a lot of reduction in shoot weight, either because the cutworms are eating more of it or because those grasses aren't able to recover as well from cutworm feeding. Then we did a similar thing with white grubs. Again, here we looked more so at turfgrass species and weed species because of the fact that white grubs are going to be more so a problem in roughs where you are going to see more turf grasses and you are going to see potentially weeds. And so we did very similar stuff of looking at white grubs feeding on these different turf grasses and then also applying nematodes and doing infested, non-infested controls. As far as resistance, this is both without EPN. We mostly didn't see any effect on white grubs with resistance. We actually had two species here we looked at. Once we separated them out, they had very high variability. So, it's probably why, we didn't really see much resistance here. There was some difference between white clover and tall fescue and mass Schaefer, but it wasn't much. Then with nematodes, we did see differences, actually kind of similar to the lab experiments where it seemed like Kentucky bluegrass and white clover had the highest mortality with nematodes against white grubs, even though in the lab experiment we're testing against black cutworm. That was kind of interesting to see similar results with black cutworm in the lab and then white grubs in the greenhouse. And then here, mostly we didn't really see anything interesting here where there were differences in weight between grubs. There was less root loss in annual bluegrass, but that's not really that relevant to the system considering the fact it's a weed. So, the tolerance of a weed isn't really that important. We'd more so want to see differences between turf grasses. So ultimately, the two important points was, that nematode performance was affected by these different plant species. White clover and Kentucky bluegrass seem to have the highest mortality with nematodes in both lab experiments with black cutworm and in the greenhouse with white grubs. And then that there were kind of these separation, even though it was a little bit messy, of cultivars and nematode performance, creeping bentgrass cultivars on black cutworm, both in the lab and in the greenhouse. It seemed like these two cultivars kind of separated out as the ones where nematodes perform best. There was not much resistance that we saw other than Kentucky bluegrass to black cutworm. But again, that was something that we already knew from research. But there were a few of these creeping bentgrass cultivars that seemed to have less shoot loss to black cutworm feeding in the greenhouse. And that mostly we saw similar results between the lab and greenhouse experiments, like I talked about, even between lab experiments with black cutworm and greenhouse experiments with white grubs. And then, definitely fairly similar, even though we're a little bit different with the creeping bentgrass cultivars in the lab versus the greenhouse with black cutworm. And so with that, I will finish up, and I'd like to thank my collaborators, my PI, Albrecht, here, who worked with me on the last few projects, our former postdoc, Terry Cool, definitely some of the other turf grass scientists here, weed scientists that work with us on the project with converting annual bluegrass weevil into a biological control agent of weeds, and then also the turfgrass plant pathologist here that worked with us with the project with synergism between fungicides and nematodes and insecticides, and then some people I worked with previously on the first project in my PhD. And with that, I will take any questions.